And here from Professor Dr. Christoph Borner, who will discuss herpes simplex virus. How does it survive in the body of so many humans? How? Here we'll explore the fascinating strategies behind viral persistence or latency. I don't know. Thank you. You.
Thank you very much for the wonderful introduction, and thanks to Alicia and Nadia for organizing this wonderful conference. Today I'm going to talk to you about our attempt to understand better a virus that is probably the most prevalent in the world, herpes simplex virus 1. It affects 67% of the human population all around the world. It's neurotropic, so it hides in the nervous system. But it can be recruited, for example, through fever or sunlight. The last 3 days I was in the mountains and I got some sun and I have some blisters. So this is what it happens. Then you get some cold sores, these typical blisters on your lips. This is not dangerous, but in immunocompromised patients, it can affect or it can lead to encephalitis, keratitis, or even neurodegenerative diseases. So the virus can become pathological. The virus enters epithelial cells, for example, and also immune cells, through mainly two receptors, the TNF-like receptor and nectin-1. It then disassembles, and then through several translation events, it forms new viruses. Now the question is, what does the virus need for effective virus spread? You would think that the infected cells should first survive so that the virus can multiply to reproduce. But this may also trigger an antiviral immune response because the cells are there and they are detected by the immune system. Or the alternative is you get the death of the host cells, but this of course can only occur after the virus has been reproduced. In this case, this would evade the immune response and the virus could become pathogenic. So what is, when we talk about survival or cell death, what are the two major types of cell death we have to look at? So there's a silent form, which is apoptosis, that is non-inflammatory. The cells just shrink and bleb, but they never lyse. And here we have basically two pathways. One is triggered by death ligands, like TNF or Fas ligand, which then activates apoptosis through caspase 8 and caspase 3. There is also a mitochondrial pathway with other stimuli that involves cytochrome C release and also caspase 3 activation. On the other hand, we have necroptosis, which is an inflammatory pathway that produces inflammation because the cells lyse. And here typically it's due to TNF and other death ligands, which on one hand can either lead to survival through NF-kappa B activation, or then can also trigger necroptosis, which is through two kinases, RIP kinase 1 and 3, that activates MLKL, which forms pores in the plasma membrane. And that's why the cells lyse. So what happens with the virus? The virus produces two proteins that can block apoptosis, ICP6 and ICP10. And it blocks caspase 8 and RIP kinase 1. So it blocks apoptosis and necroptosis. But it also stimulates NF-kappa B signaling. And probably, this is known, NF-kappa B signaling is a survival pathway. This is probably through the receptor. Now what happens when we infect with the virus? We indeed see in epithelial cells that there is no apoptosis. So no caspase 3 cleavage. This is the positive control. So the cells survive. While do they survive by NF-kappa B signaling? So in order to look at NF-kappa B signaling, we need to actually look at the phosphorylation or activation of IKK2 or IKK1, which then phosphorylates an inhibitor of the NF-kappa B that is degraded, and then the NF-kappa B is phosphorylated and acts as transcription factor. So we did this, and in fact, the virus induces NF-kappa B signaling. You see here clearly the phosphorylation of NF-kappa B. You see the degradation of the inhibitor, and you also see the induction of a target gene of NF-kappa B, which is TNF. So it clearly activates NF-kappa B. But is this important for survival? So we used an inhibitor. The inhibitor is an IKK inhibitor, so it acts here. So when you inhibit this kinase, then of course NF-κB signaling is blocked. And you actually see that with the inhibitor, you actually see that IKK is not phosphorylated, IKB is not degraded, and NF-κB is less phosphorylated. So indeed, the inhibitor works. But what about survival now? So if we inhibit, we should get now apoptosis. So we did a FACS analysis, and we looked for the phosphorylation of NF-κB, P65, and also for caspase-3, which is a marker for apoptosis. So what you see is the inhibitor indeed inhibits NF-κB. So this is P65 phosphorylation with the virus, so it nicely goes up. With the inhibitor, it's down. But there is no caspase-3 activation. So NF-κB signaling is inhibited, but there's no cell death. But we see less viruses. So you see less virus production, which means NF-κB signaling is important for viral replication, but not for survival. So this is our first conclusion. In human epithelial cells, the virus seems to induce NF-κB, but it's not important for survival, but for viral replication. So what happens later, after, let's say, 24 hours, 48 hours? Because we see that, in fact, we get the blisters, so that the cells, the keratinocytes, for example, they lyse. And indeed, what we find after 24 hours is that the cells undergo now a form of cell death. So you see that the cells detach from the plate, and we can actually measure cell death. So first they survive, after 24 hours they start to die. And we found out this is by necroptosis, because we see that there is phosphorylation of RIP kinase 3, remember this kinase that is important, and MLKL, which is the kinase that forms the pores in the plasma membrane. And when we take cells that are knocked out for RIP kinase 3, we do not see cell death, which means that it is necroptosis. And we can actually do the Western blots where we see that MLKL phosphorylation, RIP kinase 3 phosphorylation, MLKL phosphorylation is largely diminished in these knockout cells. So our second conclusion is, initially the cells survive. After 24 hours, they undergo necroptosis. And this is probably what we see when we see the lysis of the cells on our lips. But is this all? We just looked at epithelial cells. What about immune cells? Because the virus also infects immune cells. Now, astonishingly, immune cells are different. As immune cells do not survive. After virus infections, they undergo apoptosis right from the beginning. And we published this recently in Cell Death Differentiation. We actually see caspase-3 activation, we see cell death, and we see that this goes via caspase-8. So caspase-8 is cleaved and activated, caspase-3 is cleaved and activated, and in caspase-8 knockout there is no caspase-3 activation, there's no cell death. And we can reproduce that in another monocytic cell line, which is THP1 cells, exactly the same result. But it's also, like we see in epithelial cells, we see NF-κB activation, because the virus, remember, requires NF-κB signaling for replication. And also here, we showed that NF-κB signaling is important for viral replication, but not for survival. We see that P65 phosphorylation goes up, but with the inhibitor it's down. There is not more cell death. Remember, they undergo apoptosis, but there's not more cell death when we block NF-κB, but there is an inhibition of viral replication. So in human monocytes, the virus induces caspase-3, caspase-8 dependent apoptosis right from the beginning, but it also induces NF-κB signaling for reproduction. So final summary, and this is really the interesting thing, human cells seem to react differently towards the same virus. In epithelial cells, such as keratinocytes, you get cell death protection, so the cells survive, but later on they undergo cell lysis in the form of necroptosis. And probably this is to allow a better spread of the virus, because then the virus comes out and can nicely spread. In immune cells, such as monocytes, the cells undergo apoptosis after the viruses are reproduced. And this might be a strategy of the virus to evade an innate immune system. So it really depends whether the virus takes the cell as the own benefit for reproduction and how it interacts with the immune system, whether it's epithelial cells or monocytes. So I think this will have clinical translational application in order to understand better a virus that is basically a virus that is globally infecting so many people. The people who were involved in this work were Daria, an Indian PhD student, Emilia, which is an Argentinian PhD student in my lab, many more people in the lab, and our collaborators is the group of Uli Maurer, who is also here, and some virologists in Freiburg and in Munich. Thank you very much for your attention.